The final discussion we need to have in macroevolution is how to map these relationships. So when we talk about mapping relationships in evolution, we actually look at mapping the genome or particular genes of the organisms and looking at that relatedness. So we call it a phylogeny. What we're actually looking at is essentially a pedigree based in molecular information. So we could look at a single gene, we could look at many genes, we could look at just mutations in genes. There are lots of options, but what we want to consider is how those genes have changed over millions of years or thousands of years. So this is giving you relationships between several different mammals, in particular focusing on carnivores. So the derived trait we would look at here would be carnivores. But when we think about their relatedness, it looks kind of like a family tree, right? This shouldn't be a surprise to you. What we're looking for is where these branches on the tree or these lines actually come together at particular points. So if I wanted to discuss how long ago sea lions and walruses were related, what we do is we find the point where sea lions and walruses come together and we simply draw a line. Now, actually collecting the data and putting it together is much more complicated than this. But when we look at this relationship, we can say, okay, it's been about 18 million years since sea lions and walruses were actually related. So at that point, about 18 million years ago, there was a common ancestor to sea lions, walruses, and seals. They're all in the same branch here, right? And that common ancestor then branched off, potentially through adaptive radiation or other speciation means, and have now given us three very distinct different species. If we look at something like this is more what you would see in an article associated with evolutionary relationships. So here I show you a relationship between a bat and a gorilla, their common ancestor being way back here. Okay, and the reason I showed you this one is because we looked at bat and gorilla in comparative morphology as far as their forelimb structures. So you see it's been a whole long time since they were related. This one doesn't give you millions of years. What it does instead is you see this little tiny line up here at the top of the screen with a 1 beside it. This is essentially like measuring mileage on an old paper map. So this little tiny line says that for every length that is that long, that little tiny line, we have 1 base pair change. Okay, so if you would measure these all out, and computer programs of course do this for you, we can look at how long these lines are. The longer the lines, the more distantly related the organisms. So we see up here something called an outgroup. See, the duck-billed platypus is all by itself. So this is something known as an outgroup. That's the group to whom we will compare everyone else, all right? And very, very, very far removed from that, way down here at the bottom, is the common chimpanzee. Okay, mixed in there, of course, human, bonobo, and many others as far as the primates go, but we are very distantly related to the platypus. Remember, the platypus is still an egg layer, so being a placental mammal as primates are in this whole group from here down are all of our primates. So looking at that group compared to our egg layer up at the top, very different, very distantly related. Now this doesn't give you any time scale, so we'd have to look at a different figure to give us more of a time scale. The idea of putting together these evolutionary trees, many originally thought about these as cladograms. Cladistics and phylogenetics are slightly different. When we look at these relationships, phylogenetics gives us the molecular background and gives us that information, but it doesn't always give us all of the fine detail that we need as far as really understanding morphological relationships and everything as well. So a cladogram is simply this graphical representation, and we've already looked at some of the carnivorous mammals, but this gives you a slightly more diverse grouping of the animal kingdom, all the way down to an invertebrate in the hagfish. So we'll look at these guys. The hagfish has what we call well, you've already talked about, a notochord. Okay. It's actually maintaining its notochord into its adult state, and we'll look at that later when we look at animal kingdom. But when we look at this tree, what I want you to think about is which two of these are most closely related. So remember, to be closely related, you have to have short branches on your tree and a fairly recent common ancestor. So which of these two do you think are most closely related? How do you know that? You can write your answer on your worksheet. So common ancestors, remember, are at the joints of those branches, and what we want to think about is that if you're going to look at these common ancestors and all of the groups of organisms following that common ancestor, we have to distinguish what characteristics are going to be shared, which ones might be lost, and which ones might be gained. Okay? So these characters we can look at simply as phenotypes, but we want to know what's lost, what's gained, and what is still shared. Okay, these will be important points for us. So the clade is actually this group of organisms, or the ends of the branches. Okay, and what we want to look at is which of these branch ends on our phylogenetic tree, which of those organisms all share particular characteristics or derived traits. Remember, derived traits are ones that we've talked about before. These are unique characteristics, but we look at many of these characteristics as being so important that they're maintained throughout different groups. So how do you know how closely related organisms are? I asked you a couple of slides ago to consider which organisms might have the shortest branches, might be the most closely related. And on our tree I gave you, we have to think now, okay, which two were most closely related? Well, I hope you picked these two. Why would those two be most closely related? A seal and a lion, two different ecosystems, two very different hunting mechanisms, two very different family structure, community structure mechanisms associated with them. So why so closely related? Well, we only have 5 organisms on this tree. This is something you have to be careful of. When you talk about organisms being closely related on a cladogram or phylogeny, it is based solely on the information that you have. So if you only have 5 organisms, the idea that a lion and a seal, which are both carnivores, we see here, carnivorous feeding, that means everything after that is a carnivore. So both our lion and our seal. Other ideas on here, this tetrapod structure. So that's suggesting that the hagfish does not have a tetrapod structure. What does tetrapod mean? Tetra is 4. Pod is what? We've seen it before. Feet or legs or limbs. Okay, so this 4-limbed structure, a lizard, a deer, a lion, and a seal certainly have 4 legs or 4 limbs associated with them. So our tree holds in that everything after that tetrapod structure designation has a tetrapod structure. Same with mammary glands. A lizard does not have mammary glands, neither does a hagfish, but a deer, a lion, and a seal do. Okay, so the closely relatedness comes from this idea of short branch lengths. The derived characters, keep in mind everything after those lines has to have that characteristic. If it doesn't, we have to have an explanation, and we would have to put in a new indicator of what's going on as far as that particular organism. So this idea of a phylogenetic species concept ties in with morphological and embryological concepts that we've talked about before, ecological and so on. The idea is trying to better understand these relationships, okay? So looking at the differences, we want to make sure that our cladistics is focusing on the evolutionary relatedness, but when we actually look at systematics and bringing everything together, we're going to use phylogenetic evidence, so molecular or gene evidence, okay? But we're also going to pull in other information that we know. So maybe we know something about morphology, maybe we know something about habitat. These sorts of things are important when we really start looking at putting together a big picture, full-on understanding of the relationship of these individuals, okay? So when we map out in a classical view of tetrapods, we always put birds outside, mammals outside, and then reptiles in this big mix on the far side, suggesting that the ones to the left here are older evolutionarily, okay, versus younger on the right side. Now, if you actually map this out with all of the molecular data and map this out with our understanding of the relationship of reptiles and so on, we no longer look at age from side to side. That's not the concern anymore. What we now want to look at is where in time and morphology and all of the evolutionary characteristics do these things tie together. So we know that dinosaurs and birds are very closely related. We've looked at evidence of that before. We will look at it again. But what we'll then think about is how are mammals so far to this outside? And I'm not concerned about left versus right in this tree, remember. That's not our concern. What I'm more concerned about then is this understanding of things like conserved genes, okay, habitat structure, and so on. So this is taking more of a full view of the relationships versus a very limited view that we look at in some of our classical basic morphology understandings. So with macroevolution, remember we have to talk speciation events. So these are big events. Okay, we want to look at relationships between organisms, and we want to look at kind of big picture relationships. So when we really want to understand how things are related, we have to take into account more than just one particular thing. You'd have to take into account habitat, morphology, molecular data, everything that's available to you to really understand how organisms are related to the best of our ability currently.